Okay, we'll continue talking about techniques for nucleic acid protein identification, which involve the in vivo analysis of the epigenetic DNA-protein interaction. We have seen in the last lecture how we developed small molecule inhibitors that prevented the binding of a methyl reader protein, which is a spin 1, to bind to the methylated lysine on histone protein. Today we'll talk about another example, okay? Which is S6K2. S6K2, actually, it's a kinase protein that is responsible for the phosphorylation of the ribosomal S6 protein, and hence it promotes the ribosomal assembly to undergo a protein synthesis. It was found that S6K2 can bind to the chromatin and induce the phosphorylation, since it's a kinase, induce the phosphorylation of histone 3. So we will see here how they managed to detect this modification and prove that S6K2 is responsible for this modification. So let's start first with some bioinformatics. So initially, if you wanted to experiment something, especially with the epigenetics and the modification and the post-translational modification, you need to predict first bioinformatically if there is a site for the phosphorylation or not. So normally the site of phosphorylation on histone 3, or the site that this kind of kinase S6K2 or this family of kinases need specific nucleotide sequence on any protein to undergo a modification. So let's see how it was predicted, the site before doing any experimental work. So generally, the histone, if you look here, you'll see that this is the amino acid sequence of a protein. You can see that there is a specific 2 arginine, R, and 1 T, which is thiamine. So generally, the histone 3 consists of 2 regions, a tail region and the globular region. Actually, this is a general structure of all histones: a globular region, a bulky region, and the tail region. And most of the histone modifications occur up till now have been discovered on the tail region, in the tail N-terminal region. So predicting a site that will be phosphorylated in the globular region will be a breakthrough, since it will be the first site to be identified inside the globular region of the histone that is phosphorylated. So actually it's a T45. So what's meaning of T45? It means that it is a thiamine in the position of, it is the 45 in sequence in the amino acid sequence of that protein. And if you look closely, let's have a pointer. If you look closely for the T45, you'll see it's located here, and it has a specific sequence, and this sequence, it's already known that this specific sequence, not Y, not P, not G, the existence of R, XR, XXT is a specific sequences that kinases need to phosphorylate T need to read this sequence in order to sit on this protein to do phosphorylation. As I said, this consistent sequence phosphorylation motif, so we call it motif, it is RXR-XXT/S. So these kinases have the ability to phosphorylate either T or S, but it needs a very important requirement that the S and T need to exist in this consecutive sequence. And the class of protein that is responsible for phosphorylation of this T and S in this sequence was found to be either AKT protein or S6K1 and 2. So let's see which of these 3 kinases are actually responsible for the phosphorylation of T, since the T which is here existing in this location. So initially, an initial experiment have been done to detect if it is already phosphorylated or not, this threonine, okay, if it's already phosphorylated. So the initial experiment showed that S6K2, not S6K1 or AKT, is essential for H3T45 phosphorylation. You can see here, in this setup, they used siRNA for S6K2, siRNA for siRNA for S6K1, and siRNA for AKT. And as you know, siRNA or short interference RNA are RNAs that is transfected inside these cells, and it used to knock down the expression of the protein of interest. So siRNA S6K1 will knock down S6K1. To make sure it is knocked down, you can see in the Western blot, there is no S6K1 here. If we use the siRNA for S6K2, you will see that there is no S6K2 when you detect for Western blotting for S6K2. And when you knock down AKT, you can see that AKT is a minimum when you do Western blot with for AKT. And of course, you need a negative control, which normally we use a scrambled siRNA, which is not specific to either of these three. So what is so actually these four setup, the four blots here, actually it is the control of the experiment. You check if the siRNA have worked or not. For actin, actually it is the control for the loading on the gel to make sure that all the samples have been loaded equally. So the major conclusion of in this setup, you can see that once you knock only S6K2, you will find that the signal of H3 phosphorylated T45 totally disappeared. That's mean the direct involvement of S6K2 in the phosphorylation of T45. Okay. So we can notice that S6K2 is involved directly in the phosphorylation of T45. So we need to confirm the involvement of S6K2, and we need to as well to identify the function of this phosphorylation, since it is the first modification to be discovered in 2014 as a modification in the globular region of histone 3. So let's have a summary on the mTOR pathway, which involved the S6K2. So initially the signal, the gross signal attached to the RTK receptor tyrosine kinase, this receptor is dimerized and got activated and autophosphorylate itself. This lead to the recruitment of another protein, which is IRS1, insulin receptor substrate 1, which upon recruitment recruits another protein, which is a PI3 kinase. The PI3 kinase is a kinase that is responsible for the conversion of a lipoprotein, lipid molecule inside the plasma membrane, which is PIP2, and phosphorylate PIP2 to be BIP3. BIP3 activate BDK1, which in turn activate AKT, and AKT normally inhibit the complex formation of TSC1 and TSC2. So what is the function of TSC1, TSC2? Actually, TSC1, TSC2 inhibit a GT-based protein called Reb that activate one of the major proteins in the cell, which is called mTOR complex 1. Mammalian target of rapamycin complex 1, which in turn, this one, once activated, it will activate the family of protein of S6K, which include two major isoform, S6K1 and S6K2. It was found that there is another protein called the NF1. This is involved in learning disability disorders in children, and this protein inhibit B10. What about B10? B10 is a phosphatase on chromosome 10 that inhibit the action of PI3 kinase. It was found as well that the TSC1, 2, and B10 is involved in autism and epilepsy. It was found as well that Reb is involved in mental retardation and autism, any abnormality or mutation or dysregulation. So what's mTOR? mTOR is actually the mammalian target of rapamycin. That's the name and initials. It was found that it is involved in pediatric brain tumors like gliomas and macrocephaly and epilepsy as well. So most neurons with hyperphospho-tau, important protein, which upon hyperphosphorylated in humans, hippocampi, display weak or no signal of phosphorylated S6. So what's S6? As we said before, S6 is a ribosomal subunit that participate in the formation of the ribosome. So it is heavily involved in the protein synthesis. So the phosphorylated S6 is not associated with pathological tau, but rather a feature of unaffected neurons. So again, S6K, whatever 1 and 2, their main substrate is the S6 subunit of the ribosome. So let's have a look on the our experiment and how we prove the involvement of S6K2. Generally, S6K2, if you look closely to the domain organization or the different domains in this protein, you will see that it have a kinase domain here, and this is the location of the amino acid sequence of that protein. You have a kinase domain, you have a kinase extension domain, you have an autoinhibitory domain, you have a nuclear localization signal domain. What's meaning of NLS? Any protein that have an NLS sequence, that means at some point during the cycle of that protein, it will go to the nucleus, because this sequence will recruit this protein inside the nucleus. And once you know that the protein have a sequence that is recruited to the nucleus, you need to understand that it will have a major function in on DNA. And this is the first clue of involvement of S6K2 to the phosphorylation of histones, because we know now that it is already can be inside the nucleus because it have a nuclear localization sequence. So let's see, and let's do a first experiment to see if already and to prove if S6K2 is inside, can be inside the nucleus. So we do something called the cytoplasmic something called the fractionation, or cell fractionation. You lyse the cell, you separate the cytoplasmic part from the nuclear part. Here in this experiment, we overexpressed the two kinds of S6K2. We overexpressed the S6K in cells. We overexpressed the S6K2 wild type, and we overexpressed the S6K2 NLS mutant. So this form of S6K2 will lack the NLS sequence. So you can see when you do nuclear, when you do fractionation, you will find that the wild type majorly inside the nucleus. And when you do mutant that lacks the NLS, you will find it will stay in the cytoplasm and it will not recruited inside the nucleus. That proves that the NLS, which exists in the protein, is the major contributor for the translocation of the S6K2 inside to the nucleus. What about lamin and tubulin? Lamin, actually, it is control. It is the control for actually the fraction used in this lane is from the nucleus, because lamin only exists in nucleus, while beta tubulin is the control for that fraction to be in the cytoplasm. So let's do a kinase assay. So what's a kinase assay? Actually, kinase assay, you do in vitro, you bring the enzyme and you bring the substrate. You include with them radioactive ATP, because actually the phosphorylation, the kinase takes the phosphate from the ATP and mark the protein, the substrate. So let's do in vitro experiment first, because maybe the S6K2 is inside the nucleus, but it never meets the histone. So we need initially to see if they are in vitro, can meet and do the action. So again, it is this one is in vitro kinase assay, in which we include in a test tube, we include the enzyme S6K2, the substrate, the ribosomal proteins, and we can include as well histone. We need to check if it's phosphorylated or not. So let's see the setup. And of course, we used the two types of protein, either the wild type or the NLS mutant, which lacks the nuclear localization sequence. And you can see here, both of them have managed to phosphorylate S6 protein. That's mean the mutant, so that's mean that the activity of that kinase is irrelevant to the NLS, so it can phosphorylate S6 protein, which is very obvious because actually S6 protein is inside the cytoplasm. It doesn't need to be recruited, doesn't need the kinase to be recruited into to the nucleus. So what about histone 3? We found that the S6K2 wild type, the normal, managed to phosphorylate H3, and that was lacking for the mutant. And this was the first clue that the phosphorylation needs the translocation of the kinase from the cytoplasm to the nucleus. With surprise, we found the band here that we don't know up till now what is this, that is phosphorylated with the wild type and not phosphorylated with the mutant. Of course, one of the peculiar behavior of S6K2 that it can autophosphorylate itself, okay? So let's go to continuing proving this concept. So a chromatin binding assay was done. So what's chromatin binding assay? Actually, you divide the fractions of the fractions of material that exist inside the nucleus to have different to pinpoint where the kinase exists actually. So you bring the cells, you do Triton X, so 100. So Triton X is a detergent. You'll get the total. You will get a total lysate. You will get a total lysate of the total fraction, and this will of the total components of the cells, and this represented by T. Then you do a centrifugation. You will get a supernatant S1 and the pellet P1. So the S1 actually it is the non-chromatin bound material, and the P1 is a chromatin bound represented by S1 and P1. Then you do a DNase 1 plus salt, and of course you know DNase enzyme. It is an endonuclease that will break down DNA. So you'll get a chromatin bound fraction termed S2, and you get a pellet of non-chromatin bound nuclear matrix. So that's mean you get a protein, the proteins, actually the total proteins that is either bound to the DNA or not bound to the DNA. That's the S2 and the P2. So in this setup, we used the cells used initially. It was a kind of a stable cell lines called T-Rex HEC293 cells. Actually, it's a kidney cells. So these kidney cells, we generated these stable cell lines to be constantly expressing high levels of S6K2 wild type. So when you do the fractionation, this is a kind of fractionation, and you need to detect if your fractionation works fine. So you detect for lamin AC, you can see that lamin exists only for the fractions that is inside the nucleus. And you detect as well for tubulin, you will find that tubulin exists in the fractions of the cytoplasm. If you detect for the phosphorylated ribosomal S6, which actually the substrate of S6K2, you can see that these phosphorylations majorly appear in S1 and P1 fractions. And these cells, it is not treated, actually it is exponentially growing cells. So here, when you do Western blot for S6K2, you will see that they are expressed, they have a good expression, and as well the marker for the lamin, the marker for the tubulin, and as well you can see that ribosomal protein is phosphorylated. It is very obvious that you can see this ribosomal protein inside the nuclear fraction because ribosomal protein is not inside the nucleus, so it cannot be phosphorylated. So let's see here another potential clue that may aid to prove that S6K2 can phosphorylize histone. So what we proved until now, we proved that the S6K2 can be translocated to the nucleus. And they can phosphorylate histone 3, yes. In vitro, yes, we have done in vitro kinase assay. But you need to keep in mind that almost 90% of all in vitro experiment work. The problem will come when we try to reflect that on in vivo setup. So we need to detect if this phosphorylation happened in vivo. Other problem during detection, during proving this principle, that S6K2 might migrate to the nucleus, but it may do another job. It might migrate to the nucleus, but maybe it will not bind to the DNA anyway. It do another function, like interaction with another protein, like transcription factors, before the transcription factor bind to the protein. So maybe it can translocate it to the nucleus, but maybe it is not involved at all in the binding to the DNA. But we found that this protein have a very specific sequence at the end, at the C-terminal, at the end, is called KRGR, GRPGR. You don't need to remember the sequence. And this is specific sequence. If it's found in any protein, this motif is called AT-hoc motif. What's the meaning of AT-hoc motif? This sequence have a characteristic behavior to enable the protein, any protein that is having this sequence, to bind to the AT-rich regions of DNA. So what is AT-rich regions of DNA? Actually, AT-rich regions of DNA is regions that involve promoter of genes. So most around 80% of promoters of genes are AT-rich region. So we start having a clue that S6K2 may be involved in the binding in the promoter region of the gene. So it may be involved in either activation or downregulation of the gene of that promoter. When we checked if this sequence is conserved, what's the meaning of conserved? If you find a sequence, a peculiar sequence, and you found that the sequence is conserved among the animal kingdom, that gives a huge importance for that sequence. That means in evolution, it have not been changed. That means that this sequence is important for a certain function. So when you look to the amino acid sequence of the S6K2 protein, starting from mouse, rat, bovine, rhesus monkey, human, chimpanzee, horse, dog, oxen, you will find that the sequence almost 99% conserved. It doesn't it change. Okay. Okay, this slide proves that the S6K2 have an AT-hoc motif. So what's the AT-hoc motif? It is a specific sequence that it was found that is conserved among the mammalian species. And this sequence enables a protein to bind to the AT-rich region. So majorly can bind, one of the AT-rich regions is the promoters of the genes. So let's do some experiments. Let's do another in vitro phosphorylation. So we have a clue initially that S6K2 could be involved in the phosphorylation of histone 3. So we need to discriminate if it's precisely for histone 3 or it can phosphorylate other histones. So we've done in vitro kinase, another in vitro kinase assay. We used recombinant histones, and you can see that once incubated with the S6K2, you can see that the phosphorylated histone, it can phosphorylate histone 3, it can phosphorylate histone 2b, it can phosphorylate histone 2a. So what about if we used the histone 3 alone? We extracted this histone 3 from calf thymus, and we use nucleosome. So what's as a substrate? What's the difference between the nucleosome and the histone 3? So we need to know if it's can recognize histone 3 identified by itself alone, or it can be recognized in a nucleosome. Because one major problem between in vitro and in vivo, when you incubate histone 3 alone with the enzyme, yes, it can phosphorylate. What if especially with that T45, the position of phosphorylation is in the globular region of the histone. So what if it's a nucleosome, and the nucleosome, as we said before, the nucleosome is containing the core histones, contain all the histones. What if that this T45 is not accessible? Okay, so we need to prove that the histone 3, once inside the nucleosome, it is accessible to the kinase to be phosphorylated. And yes, it is accessible, because you can see a signal of histone, and obviously it's lower than the signal of the pure histone 3, which is logic, because the histone is buried inside the nucleosome, but it still can be phosphorylated. So what about the chromatin here? The chromatin, actually, it is a control, the loading control, to prove that you already ran histones on the gel. So you can see here, this is a band of histone 2B, 2A, 2B, histone 3, and histone 4. And you can see here the difference between running the nucleosome and running the histone 3 alone. For histone 3 alone, it appears on chromatin as a single band, while in the nucleosome, if you run the nucleosome, it will appear multiple band, of course, because it's different histones. So what's new in this experiment? The new in this experiment, two things, actually three things. The first thing, that histone 3 is accessible in the nucleosome, and they can be phosphorylated in vitro by S6 kinase 2. The second important clue here, that histone 4 cannot be phosphorylated by S6 kinase 2. The third clue here, that S6K2 can phosphorylate as well H2A and H2B. So let's do some in vivo experiment. So normally cells undergo a progressive activity after being stimulated. So in this setup, we use the normal cells, we starve the cells. So when you starve the cell, that's mean stop adding nutrients to the cells will be quiescent. It will not divide, it will not do protein synthesis, which is very logical, because S6K2 is involved in protein synthesis, since it's one of the its major substrate is S6 protein, which participate in the ribosomal assembly. So upon stimulation by a fetal bovine serum, FBS, it is a growth factor. It's a collection of growth factors used in cell culture. So this FBS will stimulate cells to undergo protein synthesis, and hence S6K2 will be fully active for the process of protein synthesis. In this setup, actually, you can see that S6K, it's here already. You can detect it by Western, and you can detect the phosphorylation signal of H3 phosphorylation by T45. And of course, actin is a loading control. So this was the first clue that S6K can exist correlated with the phosphorylation of T45. So the summary of this work, so S6K2 is the first kinase that we identified for the histone 3 at threonine 45. S6K2 can bound to the chromatin and in the nuclear matrix. S6K2, not S6K1 or EKT, can phosphorylate H3 at T45 in vitro and in vivo. When we disrupt S6K2 nuclear localization sequence, it will affect dramatically the phosphorylation of histone 3 to T45. This phosphorylation was found to be correlated with, as you remember, I told you from the first in the first lecture, we need to find the function or something near the function for this phosphorylation. And the last setup experiment identified that this phosphorylation is correlated with the activation of S6K activation in response to growth factors, which is the FBS we used. So this work was published in Journal of Cellular Biochemistry, and I was lucky to be one of the authors of this